Welcome to A Closer Look. I'm Robert Signer, Deputy Director of the Sanford Stem Cell Institute Discovery Center and Associate Professor of Medicine at UC San Diego. And today, we're taking a closer look at genetic medicine and artificial intelligence. But before we dive in, I have a confession. I used AI to help write this introduction. So if it's clear, well-structured, and inspiring, well, that's all me. If it's boring, rambles, or veers into weird dad joke territory, then you should definitely blame the algorithm. But seriously, that tiny moment where I sort of casually offload part of my job to AI points to something much bigger. Because AI isn't just writing speeches and helping us book hotel rooms. AI is permeating everything: how we work, how we make decisions, and how we understand the most complex system we know, the human body. And yes, it will reshape how we diagnose and treat disease. Despite all the hype, though, there's still a sense of mystery and confusion about what AI really is. We talk about it like it's magic, futuristic, and unknowable, but it's not. It's powerful, yes, but AI is only as smart as the data we feed it. It learns from what we give it, and in biology, where the data can be complex, noisy, and incredibly personal, that's especially true. If we want AI to help us solve real problems, to understand disease, to guide treatment, to prevent illness, then it needs to be trained on the right kind of information. Today, we're not just talking about AI. We're talking about how AI meets one of the most powerful revolutions in modern science: genetic medicine. Because here's the real story. We're entering an era where we don't just treat symptoms, we treat the root cause of disease, where we can read our DNA like a book, and in some cases, edit it like a Word doc, where we can diagnose rare diseases by scanning a genome and fix broken genes to rewrite a person's future, where we can predict your risk for disease long before they strike and maybe prevent disease entirely. This isn't science fiction, it's happening right now. This is precision medicine, and it's built on a foundation of data with insights that no human brain could ever fully decode on its own. We need to know how cells regulate genes, how RNA carries messages, and how proteins fold, interact, and fail. We need detailed maps of the inner workings of cells, and we need those maps to be readable by humans and machines. That's where AI becomes the partner we've never had before. A tool to find patterns in chaos, reveal hidden connections, to sort through billions of data points, and to tell us what really matters. But again, the output is only as good as the input. And our speakers, they're not just using AI, they're building the world it learns from. They're generating the molecular blueprints, the biological language, the raw material that allows AI to be smart in medicine. Without their work, AI is just guessing. With their work, it becomes a tool for insight, precision, and impact. They're giving AI the power to be precise, personal, and truly transformative in medicine. And if all of this feels a little bit overwhelming, remember this. Not so long ago, the internet felt confusing and scary. People thought it was a fad. They were terrified to input their credit card information into a computer. Today, we trust it to manage our money, navigate our cities, and deliver dinner to our doorsteps. AI and genetic medicine are at the same tipping point, and we're about to take a glimpse at what's on the other side. To guide us, we're lucky to have two extraordinary scientists who are helping to shape this future, not in theory, but in practice. Let's meet them. Please first welcome Dr. Yo.
I'm going to tell you a little bit about how we are uniting RNA biology with artificial intelligence in thinking about the future of genetic medicine. Okay, so let's begin maybe two decades ago, right? So 2 decades ago, your genome sequencing for the first genome would cost $3 billion. That's $1 for every base. And then in 2007, it became $1 million, which is still pretty expensive. Today, a genome is $600 to $1,000. So it's a commodity. So getting the genome sequence is no longer the bottleneck, right? Getting the genome interpreted And then that leading to therapeutics takes months to years, sometimes decades. So in the field of genetic medicine, in the field of RNA biology, we think that AI should shorten this approach, right, to get to faster diagnosis from your genome sequencing, to get to personalized RNA drugs, medicine, and then eventually to help us engineer new therapeutics that can be precisely engineered for individuals. However, as Rob had pointed out, AI needs very high value curated data sets, right? And so we will talk a little bit about that as well. Okay, so we have learned how to read and write the human genome. My lab's interested in the layer in between DNA and protein. So this is the interpreter of our genetic code. And we are hoping and developing ways to enable AI to help us do this better and more precisely. So again, simple central dogma of molecular biology. Your genes are encoded in the human genome, and these genes are transcribed into RNA, and then RNA is translated into protein. So RNA essentially translates DNA into some sort of action. It's dynamic and central to gene regulation. However, when you look at the number of genes that encode proteins in your genome, you're not much more complex than nematodes, right? So this simple C. elegans I guess you can see here, we have about 1,000 cells in the body, encode about 20,000 genes. We have 20,000 genes. We have, 30 trillion cells? So the number of genes doesn't equate to the complexity in the organism. So how do 20,000 human genes encode hundreds and thousands of proteins? So RNA comes to the rescue here. So here is an example of an RNA that undergoes splicing. It's a natural process that is present in majority of human genes, where intronic sequences or non-coding are removed from intervening coding chunks called exons. And the splicing of these exons together generate protein, right? So here you have 5 exons, 1 protein, but you could alternately splice this gene, remove one of the exons and generate a different protein. And then you can do the same for another exon and generate yet a different protein. So in this way, human cells can generate a large diversity of proteins. So then the model should be one gene equals many mRNAs from splicing equals many proteins. Well, how do you control splicing? Well, it turns out a large class of proteins called RNA-binding proteins are important in modulating alternative splicing. And actually, there are about 4,000 genes in the human genome, a fifth of it, that actually encode these RNA-binding proteins. And they don't only modulate splicing, they modulate many, many aspects of RNA processing. So the picture I want to paint is this. There's an enormous complexity in the RNA biology space in terms of data. This is critical for us to interpret diseases, develop new therapeutics. RNA-binding proteins form very complex networks, and even simple genes encode many different isoforms. So we think that the future requires us to leverage machine learning, deep learning, AI, to help us interpret the data and then develop ways to generate new hypotheses about biology. So just for the folks that may not know this, RNA is a gold mine for therapeutics, right? As I already mentioned, 20,000 coding genes are present in the human genome. Only about 500 have been drugged so far. But with alternate splicing and other RNA steps, you have about 100,000 over RNA transcripts. So RNA expands the druggable genome. RNA is also programmable. As you'll hear later, RNA can be programmed to bring proteins to specific places in the DNA and RNA. RNA can be programmed to generate vaccines. RNA can be programmed to generate proteins to replace proteins that are missing in individuals. But RNA can also be drugged directly. And there are great examples of drugs that target RNA, like siRNAs, ASOs, aptamers, even small molecules, right? And so RNA is really a rich place for therapeutics. And for those who don't know, RNA is also a problem in many diseases. So we work in neurodegeneration, ALS. We've worked in Huntington's and myotonic dystrophy. We've made some inroads into oncology as well. And defective RNA processing and biology underlies many of these diseases. Here's one example. This is not from my group, but from Adrian Krainer's group, which is a very good example of one, an RNA-binding protein SMN is missing in patients that get spinal muscular atrophy. Now, to fix the disease, there's a backup copy in the human genome called SMN2. If you fix the splicing of SMN2, you can restore the functional protein SMN. To fix the splicing, an antisense oligonucleotide that targets the RNA can be used to block another RNA-binding protein in green from binding and suppressing the exon. So here's a RNA-binding protein disease that can be fixed by blocking an RNA-binding protein from binding an RNA with an RNA-targeting drug, right? And so this is one example, but is now the basis of many, many examples. But how do we identify these important RNA-binding protein-RNA interactions? Well, you have many RNAs in the cell. You have 4,000 RNA-binding proteins. They all interact with different RNAs. How do you know which one binds where and exactly the nucleotide that we have to then design a new drug on? So we have been generating very high-quality data sets with technologies we have developed and improved over the years. One is this enhanced clip approach. There are many complex steps here, but essentially you get to a point where for a given RNA-binding protein in green, you can identify with sequencing, the same sequencing you use for genome sequencing, the binding site for where it sits on the given transcript. We have looked at maybe only 3% of all the RNA-binding proteins that exist in the human genome and mapped their binding sites on RNA in a variety of different cell lines. And the first lesson is quite simple. It gets even more complex, right? So each RNA-binding protein binds to a different place. So in red, this RBP binds to coding regions of transcripts. Others, like in green, RBFOX2 binds to deep intronic regions or intronic regions around exons. So everyone looks different. Without this large data set, it'd be very difficult to then make conclusions about anything in RNA biology. We've been improving these methods to use RNA editing enzymes that modify RNA, not DNA, to tell us where these binding sites are at single cell level resolution. So RNA and AI is actually a powerful combination. Here we have used deep learning and tools to predict new RNA binding proteins on the left, or predict vulnerabilities in the binding sites for other RBPs in different diseases, muscular diseases and so on. We've also been able to use AI with imaging to identify compounds or genes that affect protein aggregation in neurodegeneration. These are some of our published papers. And so what we hope to do is eventually gather a lot of these to generate new RNA therapeutics, right? So can we extract information about the genome or the RNA binding protein, RNA binding sites or the proteome? Can we engineer these with rational design or high throughput assays? And can we equip these proteins or RNA with elements that enable us to convey a particular function on the given RNA substrate? In the lab, we also do a lot of stem cell models for neurodegeneration and other diseases. And then you can test these therapeutics in these different models. And we have good examples, but we don't have time to discuss today. But I just want to remind you, there are many therapeutics already that exist leveraging design RBP RNA interactions. Antibiotics is a good example. Little compounds that bind bacterial ribosomes, it affects protein RNA interactions. siRNAs, right, is not the business end. It brings an RNA binding protein to knock down a gene. Antisense oligonucleotides brings an RNA binding protein, RNase H enzyme, to target and degrade a toxic RNA. And then the CRISPR system you'll hear more about also leverages a guide RNA, and that's the part that makes it programmable. And so we hope that with our data sets, with our AI methods, imaging, with more relevant data sets, we hope that we can generate from discovery to cures faster. I think that's the sense of urgency today, and in this climate, I think it's important to focus on collaboration and scalability. And for the most part, science is ready, the tools exist, the need is urgent. So I want to thank you for your time, and of course, thank my lab and ecosystem, somewhere here, right there we go, and then hand this over to Alexis. Thank you so much.
I am going to talk to you about the DNA side of genetic medicine. And in particular, I want to start with human genetic variation and then move on to how we can manipulate the sequence of our genomes. And so first, I just want to point out that, genetic variation exists, and it's not always a bad thing. And in particular, so our genomes are made up of, 4 bases, A, C, G, and T in a particular order. And 2 people's genomes have largely the same sequence, but they do differ in many locations. And here what I'm showing is that 3 different people differ by just the identity of this base at this position. And so this is known as a single nucleotide variant, or SNV. And so I'm going to be focusing on SNVs and single nucleotide variants. And so bringing up this idea of the central dogma again, any sort of single nucleotide variant that we have on the DNA level is going to get transcribed onto the RNA level. And if it's in the coding region of the genome, meaning that it encodes for a protein, it can cause a change in the sequence of the protein. And so it can be very, hopefully it can be easy to understand how these single nucleotide variants can impact our cells. And so while many of these variants are what we call benign or just simply contributing to our traits and making us different, some can have a negative impact on our health. One other thing that I want to mention is that these types of single nucleotide variants are the most common type of genetic variation. You can imagine 2 people's genomes could differ by a deletion or insertion of a base, but I'm, again, just going to focus on single nucleotide variants here. And so sickle cell disease is probably the most commonly known pathogenic single nucleotide variant. This is a T to A mutation in a coding region of our genome, and this is the HBV gene, or hemoglobin beta gene. And we understand this disease very well. It causes, these horrible phenotypes that contributes to the disease. And the way that we are, currently thinking about treating or actually curing this disease now is by going into the cells of an individual and changing their sequence in order to get rid of the disease phenotype completely. Right. And while many of you have maybe heard of CRISPR, I'll go through it very briefly and with a really high level. This is one of these genome editing tools that really revolutionized the field. And so, as Jean mentioned, it's comprised of a protein and an RNA component. And this RNA component allows us to kind of turn this protein into a homing device and tell it exactly where in the genome to go. And it's through these base pairing properties. So maybe you've heard of, our genomes are made up of A's, G's, C's, and T's. Well, A always base pairs with T across from it, and G always base pairs with C. And so that's how we tell this protein, where to go. Maybe the hemoglobin beta gene, for example, is that we design the sequence of this RNA to be the complement of where we want it to bind in the DNA. And so I want to point out that what I call traditional CRISPR, this is the sort of what we call the wild type Cas9 protein. It works by introducing DNA breaks. So we tell it to go to this location in the genome through that guide RNA sequence, and then it cuts the DNA. And there are lots of different ways that the cell can repair this break. A very sort of minor repair outcome is to modify the sequence in a way of our choosing, but the most common outcome is through this non-homologous end-joining pathway, which results in insertions and deletions of bases, which basically disrupts the gene, okay? And so what I just want to highlight here is that traditional CRISPR is well suited for gene disruption, but not precise correction. These double-stranded breaks can also have many negative consequences, such as resulting in large-scale chromosomal rearrangements. They're also very toxic as well. And so, as Rob mentioned, I developed a different type of genome editing agent called base editing. And what we do is we use CRISPR-Cas9. So here's its guide RNA, and here's the protein. And hopefully you can kind of visualize here this base pairing between a portion of the guide RNA and the DNA. That's how we program it. But instead of cutting the DNA, we leave it largely intact. We just nick one strand. And then what really sets this technology apart is that we engineer these enzymes that edit the DNA and actually do a chemical reaction on the DNA base. And so we have two types. And so in one case, our enzyme does this chemistry here. We take a C base and we edit it to a U base. And while a U base is not a normal type of DNA base, the cell can actually read this U base as a T. And so we can get an overall CG to TA base pair conversion with this base editor that we call the cytosine base editor, or CBE. We also engineered a different enzyme that does A-base chemistry. Okay, so it acts on the adenine base to do this chemistry. And so this is called inosine, and it is read as guanine through DNA repair. So we can take an AT base pair and convert it into a GC base pair. Okay. And since we are not breaking the DNA backbone, these processes are very efficient. And so I stole these images from Verve Therapeutics because I think it really nicely kind of demonstrates the difference between the technologies. With traditional CRISPR, we're cutting the DNA, we're disrupting it. So we're kind of like mucking up the genome in this particular location. Whereas with base editors, it's more like a pencil or an eraser and a pencil, where we can precisely go in and change one base into another. And so while these tools are very powerful and useful, and even traditional CRISPR is very useful. So the recently approved or FDA approved Vertex therapeutic for sickle cell disease actually does use traditional CRISPR. But there are a lot of ways or areas within the field to improve the tools. And so, for example, our overall editing efficiency. Okay, sometimes we don't need to edit 100% of the cells in the body, but we still do want that efficiency to be high. So there's a lot of area for improvement there. You can have off-target editing, so editing at a location that we don't want. That's obviously very bad, and we don't want that. We want to minimize that. You can sometimes have bad immune response to some of these tools because your body is recognizing it as something foreign. And then more specific to base editors is that, as you may have noticed, we can only do two types of point mutation introductions. Okay, we can only do C to T or A to G editing. And so AI can actually help a lot with helping us to engineer new tools in a more streamlined manner. And I'll give one particular example here. But so with all tools, there's lots of areas that we can change. So we can change the CRISPR component of the tool. We can change the guide RNA component as well. I'm going to focus here on the enzyme that does the DNA base chemistry. So we have tools that can do chemistry on an A base and a C base. So these are these enzymes. We actually have a decent sort of database and set of many enzymes that we know can do these chemistries. What you'll see is that in AI, we always need a training set, and then we need to use that training set in some way to give us an output. And so we can take this training set of known enzymes that can do A chemistry and C chemistry, put it through our AI bot or our robot, whatever, who can look at all these enzymes and see something that we can't see just from, looking at crystal structures of enzymes or kind of looking at the sequence of the protein. So it can find these very, the secrets, and then tell it, oh, well, now we want an enzyme that can do chemistry on a G base. And it can take, these training sets and give us a handful of enzymes to try. And so without the AI kind of helping us home in on a handful of enzymes to try, we would just be overwhelmed with, millions of enzymes to try to test in the lab to find G-based chemistry. Experiments take a long time to run, and we don't have the manpower to try a million enzymes. So one other example, so I want to highlight this amazing success story that was published a few weeks ago. If you want to read the work, it's in New England Journal of Medicine. New York Times picked up the story. There was a baby born with a mutation in a particular enzyme that's required for urea cycling. So he had no functional copies of this gene, and they estimated he had less than a year to live. Many researchers all banded together to come up with a genome editing strategy to correct his faulty gene so that he would be able to process urea. And they managed to both identify a treatment and get enough safety data on it for FDA to allow them to treat the baby. And he was actually treated multiple times, I think as early as 7 months. So that's amazing. Identifying a strategy and getting safety data on it in only 7 months. This was a success story, but there was a lot that all had to align and come together for this to happen. And so I just want to kind of touch upon the genome editing strategy development side of this. So this is a figure from their paper, so it's not super visually pleasing, but it's from them. So this is the sequence that this baby had in his genome. And so he had an A right here when he needed a G there, okay? And so they recognized that they could use an adenine base editor to correct this gene and restore function of the protein. However, as I mentioned, we have CRISPR, and it needs a guide RNA to tell it where to go. Well, each guide RNA is base pairs with a 20 nucleotide region of the genome. And here I'm just showing 7 different possible guide RNAs, okay? All differing just by 1 base pair, okay? But all 7, if we look at the sequence, any of these 7, you could even have 4 more going this way, 4 more going that way. Any of those could work. It's totally reasonable, and we really didn't have any way to know. And so for each of these 7 guide RNAs, it could be combined with over 10 different types of adenine base editors. So there isn't just one adenine base editor. There's many different variants. There's many different adenine editing enzymes that can be fused to Cas9. So if you do the math, this is already 70 different combinations, and they had to experimentally test and validate and measure the editing efficiency of all of those different combinations, which is incredibly expensive and time-consuming. And again, it literally took a village in order for this study to be completed. So one of the things that AI can help with is generating large data sets. So in advance, looking at lots of different human genome sequences, testing lots of different guide RNA base editor combinations to generate a huge data set of data looking at editing efficiencies, so that when a baby is born and it has a new sequence and a new mutation, we can go back to this data set and kind of come up with a smaller list of editing strategies for us to experimentally test and validate, especially when time is of the essence. So this is another thing that we're doing in the lab, is trying to generate these really large training data sets so that we can be helpful in the future when we really need to develop a treatment in a very short amount of time. And then finally, I want to go back to this slide and mention that, when these genetic variants are in coding regions, it's, straightforward to think about how that can, impact a protein. But a lot of these variants actually occur in other regions of the genome. And so this is a breakdown of variants associated with neurodegeneration. And actually, if you see protein coding is this green bar. So very, very actually small number of these genetic variants are in protein coding regions. And it's because of this and many other reasons that we're really bad at looking at somebody's genomic sequence, finding a single nucleotide variant, and then understanding what that's going to do. So out of all of our vast data sets of human sequencing data, about 99.5% of variants are completely unclassified. We don't know what they're doing. The less than 0.5% that are classified, many are kind of considered uncertain, so we don't understand them. And that's because, again, we're using computational methods, just kind of relying on sequencing data from individuals. And these data sets are very biased as well, because we're relying on people agreeing to be sequenced and, donating their sequencing data for these studies. And so that leads to a lot of bottlenecks in the way that we are currently thinking about interpreting genetic variation. In order to do this computationally, we need to identify variants in a large number of individuals, which just isn't going to happen. And additionally, each person has millions of variants, so it could be specific combinations of variants that are contributing to a disease. And so just relying on sequencing data from people is not going to do it. And so what us and others are doing is kind of generating data sets experimentally and in cells to understand how variants will impact our lives. And so you can think about, each one of these colors is a different mutation or single nucleotide variant that we've introduced into our cells in the lab, and we can study how it's impacting the cell there. But then there's another layer of complexity in that, as I mentioned, each person has many variants. And so now these colors are kind of representing different genetic backgrounds. So from, maybe different ethnic populations, or even, within an ethnic population, like different regions of the United States. And these variants could act differently depending on what the genetic background is that they're being introduced to. And then also thinking about different genetic variants might have different impacts in different types of cells. So you can, we look at these databases and we see, this genetic variant is causal for colorectal cancer. Well, why is it only causal for colorectal cancer? Why not a different type of cancer? And so this is because these variants behave differently across against different cells or tissue types. And so eventually we'd like to, cross all these different things, different variants across different backgrounds, across, within different cell types to generate these really rich data sets that can be used to train models to predict pathogenicity of future variants. And so with that, I'll end. And again, I just want to thank my lab as well. Thank you for your time. And, Jean and I would be very happy to take questions now.
Thank you both so much for terrific and enlightening talks. There's lots of questions, and we're going to open it up to the audience as well to get those. And so, but let me start off with a concept that sort of came up in both your talks, and that is the concept of time, right? The hope here is that this is going to speed things up, right? Make things faster, right? Jean, you talked about a pressing need. Alexis, you talked about how, in the case of a child who's got a short amount of time to live, like we are up against the clock. So can you talk about a little bit, like, what is the timeline here? Like where this is, we're talking about this one incredible case to when this becomes really part of our normal medical care, right? That AI and is really involved in that.
Well, maybe I can start. So we have a personal story. My grad school advisor, Chris Birch, has a child, and this is public, so I can speak about it, right? And the child has variants in a sodium channel gene, and it turns out that if you alter the splicing of this gene, you could technically fix the disease, right? And so in this case, the child is very lucky because Chris is a faculty and knows about RNA splicing. His wife is a faculty that can generate a mouse model for the disease, right? And then he reached out to us to generate stem cells for the patient, for the kid, and we managed to work with companies to bring in ASOs, antisense oligonucleotides, to target the RNA, and eventually everything is working out. Right, so Boston Children's Hospital, I think, is evaluating using some of these ASOs to treat the child, and there are nonprofit organizations like Ann Lorem that will do this and fund it for the lifetime of the individual. But with every disease, time is critical because the later you treat, in this case, the neuronal networks forming in the brain is critically dependent on the proper functioning of this sodium channel gene, and the longer you not treat the patient, the more damage is being done, because the patient gets epileptic seizures, sometimes multiple times in a day. Every seizure is like a rebooting of the brain. So can you decrease this faster? So I think that's one example where it still took, I don't know, maybe 2 years or a long time to get from sequence data to where's the variant to ASO design to safety, all that, right? If we can do this faster, there are many more kids that could be fixed. And I think this is the same for genome editing and same for small molecule therapeutics, right? If you can understand how the genotype, as you mentioned very eloquently, gets to phenotype, and then what is the therapeutic strategy there, and then getting safety worked out fast, I think that's critical, yeah, in that sense. I don't know if that answers the question.
And so, like, where do you think that puts us, 10 years from now, 20 years from now? Like, is this something that we're all going to experience in one way or another, or?
I think it's exponential too. Like now, working in the lab and seeing, playing around with the AI tools and seeing what it outputs, and you're like, oh my gosh, this has, so far to go. But as you said, growing up, the internet wasn't really, like for me, it just came out and we're like, oh, what is this? And then now we can't even imagine our lives without it. And so I think currently looking at the landscape, it's kind of funny to play around with, and you're like, oh, well, job security, it'll never replace me in the current form, but I think the progress is going to be exponential.
I look at AI as with most technologies, and I think the genome sequencing one's a good one, right? Where if you think about Moore's law, right, where every 2 years, every year there's a doubling of the reduction in size, so your transistor gets better, right? So the cost of sequencing, number of bases, dropped faster than Moore's law, right? And so that was amazing. I think AI is basically following that trend or faster, right, in decreasing the cost of things being done. But I think the lesson also from sequencing is that you need continuous technology development and also continuous validation of that technology with better and better data.
Yeah.
So I spoke to folks at DARPA, right, biological defense, and they all are interested in AI, but that folks recognize that actually the right data sets is actually probably the bottleneck for AI these days, because I think in biology there's still, other than genome sequencing and RNA-seq, right, there's still a dearth of good data sets that are well curated, especially with the patient phenotype metadata, and to track outcome, right? And so I think that's still going to be there. I'm not sure if that, yeah.
So, do you think we ever get to the point where we trust it enough that we don't need to validate it? That
No way. I wouldn't. I would always, the way that I think about it is build a nice data set that you know is a nice data set. So also compiling data from different studies and different labs. You don't know if those data sets were generated differently. So I'm like, generate a nice quality data set that's consistent, and that all the data points can be properly compared to each other, and then have it spit out a smaller set of things for you to experimentally validate and try. I would never say, all right, give me your number one thing. That's what we're going to do. I'm going to put this in a patient. Definitely not.
There's a phrase I like to use: trust but verify.
Yeah.
Right.
That sounds like wise advice for sure. So in that way, like, to what extent do you think that like these technologies and are going to change lab research, right? Is lab research going to sort of fall by the wayside and it's going to become, largely computationally driven, or do you think there will always be an important space for?
So maybe 2 decades ago, when I was first starting my lab, I put a premium on people that can think like an engineer, do coding, right? In some sense, almost saying if you can do computer science and engineering, you can definitely do biology. But these days, folks in the lab are already using ChatGPT and others to like write code, right? I'm flipping my mind, right? It's like thinking, well, actually, so coding or the ability to think computationally may not be the bottleneck anymore in the stuff that we do, right? Genomic medicine, it is being able to tie things together and then define creatively what is the right experiment that can generate the right data, so then you can feed into the algorithms, right? So I don't think it's going to replace like lab work, because there's always going to be creativity that will arise from accidental discoveries you can see from the work. And I don't know yet if AI and even robotics can replace that. But I see this with cautious optimism. So.
Well,
I don't know.
I'm relieved to hear that lab work isn't going to be completely obsolete here. But I guess then, thinking like from an economic perspective and jobs, and lots of people out there that are training in these areas, where do you think there are sort of new jobs that are going to emerge from this type of work? Are there entirely sort of new aspects of research and research positions and training programs that are going to be needed, to really address aspects of genetic medicine, AI?
I mean, I think everybody needs to learn how to utilize AI and to help them accelerate their work. I think, as scientists, we're constantly generating new training sets, essentially. And so I don't think that's ever going to become obsolete. And you can kind of see, as an analogy, like making cars. Yeah, you can, build a factory and have a robot build a particular car. But then what about, like, how do you design the next car, though? You can't use the robot to iterate and design and make something new. That needs to be done manually. So I think that our jobs are safe. I think everybody should utilize this tool to help them. But again, I don't think it's going to be, I think you need experimental validation and also generating new data to feed the models. So I think you need to be able to do both.
And, with genetic medicine, right, we talk, because it's genetics and genetic disorders, a lot of the times we're talking about inborn genetic disorders, right? These are, often babies, children born with things. But is there space for that? Is there space for genetic medicine beyond that, right? Like I do a lot of work on aging, right? Is there going to be space for how genetic medicine can alter human aging and lifespan and health span and those things beyond treating, sort of rare genetic disorders?
Yeah, definitely. So I think, though, that a lot of these more complex traits, cardiovascular disease, which you can kind of, consider a disease of aging, it's so much more complex because there's so many more variants that like are kind of working together to contribute to risk or to long-lived whatever. But there, I mean, even like Verve was, I think Eli Lilly bought it the other day.
Today or yesterday.
Yeah, for this particular genetic medicine that's for heart disease risk. And right now, all the clinical trials and everything, they're testing it on individuals who have particular genetic variants that make their cholesterol really, really, really high. But I think kind of like the long-term plan there is to maybe develop that as like the new Lipitor. And so like a genetic way to prolong your life by making you healthier, by tackling one of these very more complex traits.
I mean, complex diseases may still have genetic targets, right? And so I think that's the way to think about it, right? And even if you look at like things like aging, I mean, neurodegeneration, ALS, all that, there are variants that show up. And even when there are no variants, sometimes the downstream molecular phenotype is still the same, there's some convergence. So that argues that there should be genetic targets in these diseases as well, right? Complex diseases, so yeah.
Or targets at the RNA level.
Targets at RNA level.
Well, I do want to open it up to our audience for questions. So if you do have a question in our audience, please raise your hand, and there's a mic that's circulating. While we get that going, let's talk about sort of a little bit of the elephant in the room, right? And that's like the ethics of all this and sort of how these topics, I think, make people nervous, and justifiably so, probably to some extent, right? As we gain sort of more genetic information about people, right? How is society supposed to respond to this, right? This is, if you have certain risk for disease based on that, like how is that going to affect your health insurance? How is it going to affect your lifestyle, right? Like, what are your thoughts there? And like, is this something that needs to slow us down, or is this something that is helpful in the long run?
Well, I think a lot of people are talking about it already, and not just scientists. There are bioethicists who are joining the conversation, and I think that's really good. There's a lot of talk about somatic genome editing versus germline genome editing, where germline genome editing is passed on to your children, whereas somatic genome editing is not. And so there's a moratorium on germline editing. And I think the main worry and the main thing that we should think about is the cost of these treatments. And it's not fair if treatments to make you healthier and live a longer lifestyle are so expensive that only rich people can afford them. There's a lot to unpack in this, certainly, and I think we could talk about this for like hours and hours. But there are a lot of concerns, but I think the field is moving in the right direction by just addressing them and talking about them early.
And I mean, you talk about cost. Do you think that AI is going to bring down the cost of making therapeutics, or is it or in what way can it have the biggest impact there?
Sure. We had this discussion a couple of weeks ago with a lot of genomics experts in the room, right? And we go, if we heard of Moore's law. There's the converse, which is if you flip Moore's around, it's E-rooms, right? So there's E-rooms law, right? Where actually the cost of bringing a drug cost of bringing something to patients, a drug to patients, is not going down. It's actually increasing over time. So it's inverse, right? Why is that? Technologies are getting better. We've got better and better technologies over the years. So why is the cost of bringing a drug to patients going up, right? And a large part of that is regulatory, but a big part of that is also that you're going after more and more like bigger diseases where you need, the enrollment is crazy. And then most drugs fail at safety. As far as I know, there are no good models that can predict, if I throw this in the patient, will it be toxic, right? Toxic. So that becomes a bottleneck, right? Okay, so let's say you have more and more technologies, but all these other bottlenecks are still there, right? So you could have more drugs because AI gives you more candidates, but then enrollment, clinical trial, regulatory path are all still going to be an issue. So I'm not so sure the cost across the board is going to drop. The cost for particular sets of drugs may drop because the technology to manufacture that drug drops, that modality, right? But I'm not sure if that's going to be true just across the board.
I mean, yeah, for sure. And you raised like a really interesting point. Like, are these technologies that are going to enable us to test more things? Like, you talked about like, look, we've got 100,000 potential targets here that we've never even considered, but how do we actually test that many if we get to a good point, right? Like, how do we get enough patients to get all the data that's required? Is that going to require AI to help us refine that list to the best possible choices,
I think clinical trial innovation may actually require, and this is already starting to take place, right? AI to actually enable us to, find better ways to enroll, right? And enrollment with intelligence, right? So like for example, we have a study in the lab that looks at that in neurodegeneration you have sex differences with different pathways that might be, related to, the different pathways that are affected in different sexes, right? But when you run a clinical trial with some drugs, so you just maybe make a mixed bag, then it fails. Then you go, well, actually, if you had separated this by, in this case, by sex or by gender, right? Suddenly your p-value becomes very, very good, right? Now, so that's one example, but clearly you can use the genetic information to segregate the population so that you're more likely to see that this right population can be affected by the drug. So that to me is a good way to speed up the process and get higher success in drug discovery and drug development.
Well, are people doing that, right? Like there's got to be a ton of old clinical trial data.
Yeah.
Like, can we take all of that now and put it into, use AI to say, wait a minute, maybe there are people that this could have helped and we missed it because we're not as good as the machine at seeing those outcomes.
There's some going on, but I think there should be a lot more. Yeah.
Yeah, I know. One of my friends actually worked for a company that did exactly that, like looking at clinical trial data and combining that with high quality DNA sequencing data of the people who are enrolled to try to say, okay, so these 5 people responded really well to this treatment, the 15 other people didn't. Is there anything that we can learn that differentiates those different populations of people to figure out, like, oh, this drug, maybe it only has a very niche set of patients that it would benefit, but if it cures those people, then it's totally worth it to spend the money to try to find that information.
Yeah, like how do we leverage all the existing data sets? You talk about really finding that, but there's so much data out there, and makes me want to take all the experiments that didn't work in the lab and put them into a machine and find out what we missed along the way, But
that's actually the critical piece, right? Because AI needs both positive and negative data. And actually, even when we talk about doing small molecule screens, right? Each screen, typically they tell you what's the positive hits, but we don't tell you how badly the other ones, they did not work. But actually that negative space is actually even better because it constrains the amount of choices you have in diversifying the sets of positive hits you got. So negative data is actually really important. We just don't do a good job publishing negative data.
Yeah, but
I agree.
I think it could save tons of failure for other people and now train machines to do even better for us.
There's just not a lot of incentive to do it right now, right? To publish negative data. Yeah.
We have a question.
Gene, so I was a little late for your talk, so I asked Chat what a good question would be, and it came up with this interesting one that you've already touched on a little bit. It's given your work integrating RNA biology with machine learning, what ethical or societal considerations do you think researchers need to prioritize as RNA-based diagnostics and therapeutics become mainstream? So that's an unending question. And then the second part of the question is in your lab physically. It'd be interesting for all of us who don't do lab work. Do you have what kind of AI setups do you have in your lab that would be, science fiction for us? And then the third question is, where do you get most of your ideas? So you can answer any of them.
Okay, so the AI setup is easy one, right? It's not actually, most of the AI setup isn't very interesting to look at. It's in the cloud, right? You have, the supercomputing cluster. You can order H100s in Amazon, so you do most of the computing in the cloud. I think what's interesting in the lab is what are the interesting instruments that generate the right data for us to use in AI, right? And then you said something about ethics. I think actually Alexis' slides are actually really useful, right? Because she pointed out that a lot of variants are found in the most of the genomes that we have available are European genomes or variants, right? So there's a lot of ethnicity and racial information. It's just like missing. And when we think about biomarkers for disease, if you want drugs to be leveraged for different groups in different races, we should probably collect that information so that we can develop the right biomarkers for that disease, right? And then you said something about what new.
Where do you get your
Where do your
ideas?
ideas come from?
Not ChatGPT.
No, not yet. But maybe I should try. I think a lot of it is through discussion with colleagues and friends in the field, or even like with you, right, or people that are not in the field, because sometimes interesting things come up. So we're always trying to talk to. I'm very boring at dinner parties. I just try to talk to people about ideas instead of how's the weather or the game or something. Yeah.
So we're nearing the end of our time. So I want to leave you with one last question. So thinking on your career, sort of, what gives you the most hope, right? Like where is this taking us in your sort of wildest dreams?
I think it's the cost of sequencing going down, meaning each patient can get sequenced. We can really identify the cause of a disease. The possibility of generating lab reagents like cell lines. Jean mentioned patient stem cells. So we can actually do editing in relevant cell lines and identify these sort of bespoke editing strategies for a particular patient. I mean, this was unheard of even like 5 years ago, and so I think we're just accelerating so quickly. Like we're in the logarithmic stage where exponential where it's off the charts. And who knows where we'll be 3 years from now when we talk about AI, or maybe there will be something else that takes over and is more exciting than AI.
So I have 12 high school students in my lab right now. That gives me hope.
Love that.
Yeah.
And so one of the things that gives me a lot of hope hearing both of you talk today is like we're talking about these technologies that are sort of changing the face of things, right? But that you're telling us that the most important thing for these technologies remains data and remains evidence, right? That we really are following the data, we're trusting the data. We are not just, it isn't just hope, it's hope driven by real science and real evidence. And that is, I think, the key to driving us forward. So I want to thank you both for joining us today, thank our audience for being here.